Neurons. They're small, there's plenty, and their function is essential to make us, us. But how do we know how many neurons make up our brain? Who did all the counting, and how did they do it? The truth is, many people did. Using different methods, researchers have counted, calculated, and extrapolated the neuron count of the brain for a long time. Over the years, these methods have yielded varying results, with one number rising above all others: 100 billion neurons. Pretty recently, a new figure has been announced, which is much more precise and specifies an important fact about the cellular makeup of the brain. Now, how would you go about counting all the neurons? Even if we didn't know the exact amount, surely there are too many to manually count each one. Well, yes. None of the three main strategies involves counting all the neurons by hand. However, you don't really get to avoid counting altogether. Take the histology technique, for example. Here, we count fixed and stained tissue samples under the microscope. To make the neurons countable, we have to take fairly thin slices of only a few microns thickness. However, dividing the brain into sections this thin makes for a lot of slices to count. So, to avoid spending an eternity doing that, we take only every 10th to every 20th section. Subsequently, the neuron counts of the slices that we have skipped are modeled based on the figures of the counted slices. This approach is fittingly called model-based. The second option of counting neurons by histology, namely the design-based approach, works by taking representative samples from within thicker sections and calculating the amount of the whole section by determining the cell count of the sample. Since we are interested in a total neuron count of the whole brain, we would have to part the brain into many well-defined and internally homogeneous regions, because we can only reliably estimate the amount of neurons in the tissue if all sections from this region possess a similar structure. Departing from counting cells, we can instead turn towards measuring the amount of a certain substance of a cell and calculating how many cells would have had to be present to reach this amount. The preferred substance, which also lends our second technique its name, is DNA. With our knowledge of the amount of DNA per nucleus, we can deduce the amount of cells from the tissue that we extracted the DNA from. However, the problems of DNA extraction become apparent quickly. For a precise figure, we have to ensure that all the DNA content is extracted. Any methodical flaw will result in a deviating neuron count. Additionally, we cannot distinguish between cell types and have to rely on our estimated ratio of glial cells to neurons in order to reach a neuron count. Amongst other problems, it seems that a better method for counting all the neurons has to exist. With our third strategy, we finally get to answering the question who counted all the neurons. In 2005, Brazilian neuroscientists Suzana Herculano-Houzel and Roberto Lent introduced an improved version of Nürnberger and Gordon's direct enumeration method, which they call the isotropic fractionator. The widely accepted but never proven and properly established 100 billion neurons in the human brain were promptly replaced. Herculano-Houzel recalls that her figure of 86 billion neurons in the human brain was first met with little belief and a lot of skepticism. Since then, her method and the resulting cell count were validated and are now widely accepted. Not only did she introduce a more accurate neuron count, Herculano-Houzel also found that there are similarly many glial cells as there are neurons, contrary to the assumed 10:1 or even 50:1 ratio between glia and neurons. How did they do it? How might a method so intensely called isotropic fractionator work? Actually, it's not all that complicated. Herculano-Houzel describes it as something like a cooking recipe. Let's take a look at the simplified process. Fixed brain tissue is dissected piece by piece. Each section is put into a tube together with a solution of sodium citrate and Triton X, and is then mechanically ground until the tissue is fully dissolved in the solution. During this process, most parts of the cell are destroyed and mashed, but the nuclei stay intact. Afterwards, a DNA-specific dye called DAPI is added to the solution, which makes the nuclei visible due to its fluorescent property. Since we want to single out neurons and not count glial cells and others as well, nu-n is added to the mix. Nu-n is an antibody that specifically binds to neurons. Through centrifugation, the antibody-antigen couples are extracted and then counted under the fluorescence microscope. Not only is the isotropic fractionator implementable without heavily specialized equipment, it is not dependent on the homogeneity of single brain regions and thus allows for the easy analysis of a whole brain. So, let's recap. Many people tried to count all the neurons, yielding imprecise results due to faulty methods. Suzana Herculano-Houzel introduced the successful isotropic fractionator in 2005 and published a new neuron count in 2009, landing at 86 billion neurons in the brain.